Today we will discuss a model question paper 19, which has sent to you already in WhatsApp group. There 2 mark question, 3 mark question and 5 marks questions it is given, that is 2 marks 6, 3 marks 6 and 5 marks 1 question it is given. Okay, so I'm not showing the question paper here. We will just see the questions and how we have to answer for those questions. Okay, so in question number 1, that is model question paper 19th question paper, in that first question 2 marks question it is asked, what roles do enzyme play in detergent that we use for washing clothes? Are these enzymes produced from some unique microorganism? See here, page number 183, microbes in human welfare chapter, there is a sentence which is given about the enzyme which is used in detergents, that is lipases are used in detergent formulation and are helpful in removing oily stains from the industry. So the enzyme which is used in detergent, that is lipases, that is used for removing the oily stain. And there is another question they asked, from which microorganism it is isolated? It is from the Candida lipolytica. Okay, that is not given in our textbook. So this is the organism from where it is isolated, Candida lipolytica. This is a microorganism that comes under fungi. Okay, so this is the complete answer for this question. So you'll get 1 mark for lipase and oily stains and Candida lipolytica. For that 1 mark it will be given. So the key points of this one is the enzyme name as well as for what purpose it is used and from which organism it is getting. The second question, that is 2 marks question from the same question paper, it is asked with one choice. Okay, here all the choices it is given internally, so one question with its choice it is given here, that is second question. Why a secondary immune response are more intense than the primary immune response in human? The question is why secondary immune response is more strong as compared to primary immune response. So here first we have to see what is primary immune response. When our body encounters a pathogen for the first time, it produces a response that is called as the primary immune response. Now the same pathogen, when it is coming the next time, our body will be kept some memory of it. So the primary response, if it is showing low intensity, the secondary response means the subsequent encounter with the same pathogen elicit a highly intensified secondary or anamnestic response. This is ascribed to the fact that our body appears to have memory of the first encounter. So here the question, when you are answering, you can just say, when our body is encountering a pathogen first time, the immune response it will be with low intensity, and the same pathogen when it is encountering to the body, we can see the memory of the first infection it will be kept in our body, and the memory it will just give a high intensified secondary or anamnestic response. So that memory of the first encounter, that gives the high intensified anamnestic response or secondary or anamnestic response. So that is the correct answer for this question. Okay, and its choice is given, what is colostrum? Why are breastfed babies likely to be healthy? So first question it is, what is colostrum? Colostrum is the first yellowish milk secreted during the lactation by mother. And why are breastfed babies likely to be healthy? That is the next question. So for that, colostrum consists of an antibody IgA that will protect the infant from the diseases. That much is enough for that question answer. Okay, first you have to write what is the colostrum. So first milk during lactation from mother, that is colostrum, that is yellowish fluid. And why are breastfed babies likely to be healthy? IgA antibody present in it that will give resistance capacity to the baby. So there, that first milk during the lactation days, that is one key point in that. And another one, it is IgA antibody and resistance capacity to the infants. That is the next key point in that. Okay, next question, 2 marks question again. How are recombinant vectors are created? Why is only one type of restriction endonuclease required for creating one recombinant vector? This is from biotechnology principles and process chapter. Okay, so how are recombinant vectors are created? Recombinant vectors are created by recombining the gene of interest with the vector with the help of restriction endonuclease and DNA ligases. Now, why is only one type of restriction endonuclease required for creating one recombinant vector? See, when cut by same restriction enzyme, resultant DNA fragment have the same kind of sticky end, and these can be joined together by DNA ligase. That is the correct answer for the second question. Okay, how are recombinant vectors created? By recombining the gene of interest with the vector, recombinant vectors are created. This is done with the help of restriction endonucleases as well as DNA ligase. When we are cutting this with the same restriction enzyme, it produces same type of sticky end which can join by DNA ligase. Okay. Next question, fourth question, 2 marks question it is. State the role of UV light and ethidium bromide during gel electrophoresis of DNA fragment. First we will see the role of ethidium bromide. Ethidium bromide is used for staining the DNA. Ethidium bromide will intercalate in between the two strands of DNA and it gives a bright orange colored band of DNA. This colored DNA will not be able to visualize in the normal light. It can be seen only under UV light. So that is the role of UV light. So UV light, it is used for observing the stained DNA. The DNA is stained with the help of ethidium bromide. Ethidium bromide intercalate in between the two strands of DNA and give a bright orange color. Okay. Next questions, 2 marks question again. How are mammals living in colder regions and seals living in polar regions able to reduce the loss of their body heat? Mammals living in colder region and seals living in polar region. Right? So that is given in the chapter Organism and Population. There we can see mammals from colder climate generally have shorter ears and limbs to minimize the heat loss. This is explained by Allen's rule. Okay, and in the polar seas, aquatic mammals like seals have a thick layer of fat or blubber below their skin that act as an insulator and reduces the loss of body heat. So this is the way we can give the answer for a question. Mammal in the polar region or cold climate, there the ears and limbs will be shorter. That is Allen's rule. And mammal like seal, which is present in the aquatic habitat, they will be having a thick fat blubber below their skin that will act as an insulator that will reduce the heat loss from the body. Now next question, B question. If a population of 50 paramecium in a pool increases to 150 after an hour, what would be the present growth or birth rate per individual per hour for the same population? So here they have asked the percentage growth or birth rate. They have asked percentage growth. Okay, so here 50 paramecium, it has increased to 150. So we can just write for 50 individual 100 progenies as produced. Right? So per individual 2 progenies are produced, 2 per 1 individual. Right? So they asked the percentage, right? So we have to see how much percentage or how much per 100 individual. So 2 per 1 individual means 200 per 100 individual, like that we can say, right? So here 200 percentage, that is the correct answer for this. Okay, so if they are just asking the birth rate per individual, they are asking birth rate per individual, that is 2 per 1 individual, and 200 percentage growth it is showing by this organism, we have to say. So if a population of 50 paramecium, actually the population has 50 paramecium in a pool, that increases to 150. That means 50 additional 100 it came. So 50 members for that 100 progeny produced means we have to just say 100 divided by 50. That is the birth rate. Per capita it became 2 per per one individual, like that we can say. So 2 per one individual means 200 per 100 individual. So birth rate simply per individual, if they are asking, that is 2 per one individual, and they have asked what would be the percentage growth also. So it will be 200% growth. Okay. Next question, sixth question. This is also 2 marks question. What are transgenic animals? See, transgenic animals are the animals that can produce certain biological product, some useful biological product, and it is created by introducing the protein producing gene, useful protein producing gene we can say. Or we can say transgenic animal that produce useful biological product can be created by introduction of a portion of DNA which code for a particular product. So you can just write like this: transgenic animals are the animals which can produce certain useful product which has created by introduction of a gene for that particular product. Okay, an example like that you can just write human protein alpha 1 antitrypsin, that is used for treating the emphysema. Now, how was the first transgenic cow found to be more useful than the normal cow for human? First transgenic cow which produces the milk which contain human alpha lactalbumin, and that is nutritionally more balanced product for human babies than the natural cow milk. Okay, so that point it has to come like the human alpha lactalbumin containing, which give more balanced diet to the baby, that has to come. And after that, if you want, you can just write Rosie is the first transgenic cow that produce human protein enriched milk as alpha lactalbumin 2.4 gram per liter, like that. If you want add, actually if it is not added also there is no problem, you can just write the milk which contain the human protein, that is alpha lactalbumin, which is giving a more balanced diet to the baby, like that you can just write. Okay, so that is the answer for this question. So what are transgenic animal means? The animals which is able to produce some useful product, and that product that is produced due to incorporation of certain genes to gene into it. Okay, or we can just write by seeing this sentence how you are able to construct the sentence, according to that you can write. Okay, and its example is alpha 1 antitrypsin, like that you have to finish it off. Okay, now next question is, why is pro-insulin so called? How is insulin different from it? Next, the choice it is given, why is pro-insulin so called? See, pro-insulin is the inactive insulin produced in mammals including human beings. How is insulin different from it? Pro-insulin consists of 3 polypeptide like A chain, B chain and C chain. When pro-insulin get mature as insulin, the C polypeptide will be removed and A and B it is joined by disulfide linkage. Okay, that is the answer for this question. Why is pro-insulin so called? It is inactive form it is synthesized, that is reason it is called as pro-insulin. And how insulin is differ from it? Insulin has only A and B peptide, but pro-insulin has C peptide also. When pro-insulin from where C peptide is removed, the insulin, active insulin will be produced. Next question, question number 7 it is, write the, this is 3 marks question. Okay, write the importance of measuring the size of a population in a habitat or an ecosystem. What is the importance of measuring the size of a population? What we can understand if we measure the size of a population? That is the question. See, size of the population that tell us about the status of the habitat or the status of that particular population. See, the outcome of competition with the another species we can understand by seeing the status of that population or measuring the size of the population. Then another thing which we can find it out, that is the if any predator or the effect of pesticide application, if it is there, we can evaluate them in if any changes in the population size. So the size of the population, if we are measuring, we can understand any interspecific competition is occurring, any predator it is a prey relationship, it is in an imbalanced way it is happening, as well as the pesticide of application, its effect we can understand from it. Okay, next, explain with the help of an example how the percentage cover is a more meaningful measure of population size than more numbers. You see, the the percentage cover of biomass is more meaningful measure of the population size that is clearly explained with an example: banyan tree, single banyan tree and 200 carrot grass or Parthenia hysterophorus plants. See, single banyan tree, it is huge and it gives a large canopy, but this 200 carat grass, even though it is more number, it gives a very small, it cover a very small area. That is a best example to show the percentage cover is a more meaningful measure of population size. Next question, seventh question choice it is. Actually, I have not taken the graph here. This same graph it is given here. Okay, study the graph given below and answer the question that follow. So they have given the population density here, time here, and they have given this is A graph and this is B graph, and this dotted line is also given. They have not given dn by dt is equal to rn into k minus n divided by k like that. It is not given. That other equation dn by dt is equal to rn also it is not given. Simply the graph alone it is given without any formulas. Okay, so we will see the questions they have asked related to this graph. First question, write the status of food and space in the curve A and B. Curve A, actually it is showing an exponential growth. So there food and space, it is abundantly seen, it is unlimited. So here food and space in A graph it is unlimited, and the status of food and space, that is limited in B graph. It has some limit for the food and space in B graph that show logarithmic growth. Next one, in the absence of predator, which one of the two curve would appropriately depict the prey population? In the absence of predators, which one of the two curve would appropriately depict the prey population? If there is no predators, prey population will be increased. If there is no predators, prey population will be increased. So here what we have to consider, they have not given the status of the food and space, right? They have just given the absence of predators for a particular prey population. So here we can say if it is growing like the exponential growth, when the prey population is increasing, they can undergo intraspecific competition, means the competition in between the organisms of the same species itself. So what happened there, they will starve for the food, and we can see at a particular level when it reaches, the nature has a carrying capacity. Even though they do not have predators, they will not be able to undergo large multiplication because of this carrying capacity of nature. So we will be able to see a sigmoid curve in the next case, because predator, when it is not present, prey population will be increased in an unlimited way, and nature will not be able to support them in an unlimited resources. Because of that, they will just show the logistic growth with lag phase, log phase, stationary phase like that. Okay, so B answer is the B graph. The absence of predator, which one of the two curve would appropriately depict the prey population? That is the B graph. Next one, time has been shown on x-axis and there is a parallel dotted line about it. What is the significance of this? See, this parallel dotted line, it is showing the carrying capacity of the nature. So here the answer it should be like this: write the status of food and space in the curve A and B. Food and space in A, it is unlimited, B, it is limited. And in the absence of predator, which one of the two curve would appropriately show the prey population? That is B curve. Because when you are writing B curve, you just give an explanation like, in the absence of predators, prey population will increase and they will undergo intraspecific competition. So they will show the logistic growth. Next, time has been shown on x-axis. There is a parallel dotted line above it, but the significance of the dotted line, that is nothing but it is showing carrying capacity of the nature. Okay, that much the 3 points you have to write. Each point carries 1 mark. Okay. Eighth question, it is 3 marks question. So it has 2 sub questions. Okay, we will see that. Name a drug used as an effective sedative and painkiller. The the drug which is used as an effective sedator and painkiller, that is morphine. So here it is given morphine is a very effective sedative and painkiller. Now, for helping patients to cope with mental illness like depression, but often misused. So the second they have given, name a drug used for helping patients to cope with mental illness like depression but often misused. See, barbiturate, amphetamine, benzodiazepines, these kind of drugs, it is normally used as medicine to help patients, but it is abused as a drug. So here the second one in a question, you have to just write a barbiturate, amphetamine, benzodiazepine, any of one you can just write. Okay, now B question, how does the moderate and high dosage of cocaine affect the human body? See, cocaine, when they are taking in a moderate dose, it has a potent stimulating action on the central nervous system. It produces sense of euphoria. Euphoria means over happiness it is, and increased energy. Excessive dosage of cocaine cause hallucination. That is enough. How does the moderate and high dosage? Moderate dosage, it will just stimulate the central nervous system, produce a sense of euphoria and increase energy. And excessive dosage, it lead to hallucination. Okay, sorry. So here A question carries 1.5 mark, and B question also carry 1.5 mark. This is also a 3-mark question, 9th question. Draw a schematic diagram of a segment of a vector and a foreign DNA with the sequence of nucleotide recognized by E carbon. So here vector DNA, it is given exactly like that in our textbook. Okay, vector DNA they have just shown where GAA TTC recognition sequence they have shown, GAA TTC recognition sequence they have shown. Now, when it is cutting with eco R1, the recognition sequence is GAA TTC, and it will cut in between G and A, we know now. So G and A, here you have to show a cut, and here also you have to show a cut, and you just put a zigzag like structure here in such a way we can understand it is going to get separated. Then after that you have to show in the next step this much AATTC you just take out, and remaining thing you just keep here, and from here you just take out this AATTC, remaining you just keep here, and you just put an arrow mark from here to here, it will come and join like that. So in the next one you just join this AATT, it will come and join with TTAA, and it will be ligating each other in the presence of the ligase. So DNA fragments join at sticky ends and recombinant DNA is formed. So they are asking, draw schematic diagram of segment of a DNA vector, segment of vector and a foreign DNA with the sequence of nucleotide recognized by EcoR1. That is this one. Then draw the vector DNA segment and foreign DNA segment after the action of EcoR1. After the action of EcoR1, you have to draw this one, EcoR1, and label the sticky end produced. So here this much only they have asked. The sticky ends which is produced is this one. This is the sticky end, this is the sticky end. This much only they have asked. The third one they have not asked. So this will be carrying 1.5 mark, this also will be carrying 1.5 mark, like that. Next question, that is 3 marks question. How does the gene therapy help patient with adenosine deaminase deficiency? So here, what is this gene therapy? If a person born with a hereditary disease, we can correct that hereditary disease by gene therapy. Gene therapy, in that a collection of methods are used for the correction of gene defect. Okay, here the genes will be inserted into a person's cell or tissue to treat the disease. Correcting the genetic defect is in this way by incorporating a normal gene into an individual or embryo. So in adenosine deaminase deficiency, adenosine deaminase is an enzyme which is very crucial for our immune system. So here this adenosine deaminase deficiency, to treat, the first step it was taken, they isolate the lymphocyte from the blood of the patient, and they were grown in the culture outside the body. and we can see this functional DNA for the adenosine deaminase, that is complementary DNA. With the help of a retrovirus, it was incorporated into lymphocyte, and lymphocyte was given back to the patient's blood. As these lymphocytes and all, it is not immortal, they will undergo death. Because of that, the periodic infusion of such genetically engineered lymphocyte, it was necessary. Because of that itself, it was very difficult. So the bone marrow cell from where if we are able to the bone marrow cells, if we are able to introduce the adenosine deaminase enzyme producing gene in the embryonic stage, we can get a permanent cure in that. So here, how does the gene therapy help a patient with the adenosine deaminase deficiency? That is the question. It is a 3-mark question. So according to that, you have to give the answer. So first you have to write, if a person is getting hereditary disease, to cure that we have to do the gene therapy. Defective gene can be replaced by a normal gene. This can be incorporating a normal gene into the cell or tissue of a patient, and we can see the expression of that normal product from that particular gene. Here an example for adenosine deaminase deficiency. Adenosine deaminase is an enzyme which is very crucial in our immune mechanism. As it is not present in a patient, the first step towards this adenosine deaminase deficiency gene therapy, it was taken, lymphocyte of the patient was taken out from the blood, and it was cultured in vitro condition and incorporated the normal gene for adenosine deaminase, and it has given back to the body of the patient. As this as this lymphocyte, it is not immortal, we have to do this process periodically, means we have to do this process repeatedly. But if we are able to incorporate this gene in the early embryo stage in bone marrow cell, we can see there the normal cells which is produced from the cells which is produced from the bone marrow, they will be normal, and it will give a permanent cure. Okay, that much we have to just write in this question. Next question, 3 marks question. It is, explain the importance of biodiversity hotspot and sacred groves. Importance of biodiversity hotspots. See here, biodiversity hotspots region that has very high level of species richness and high degree of endemism. Endemism is nothing but the species which is confined to a particular region, which is not found anywhere else. So biodiversity hotspot means they will be having high degree of species richness and high degree of endemism. And what is the importance of the sacred grove, that we will see. See, the sacred grove in which we can see the all the trees and wildlife will be protected, means totally it will be protected in a particular area, that is a part of the culture. So that is sacred grove. Sacred grove where we can see large number of rare and threatened plants, large number of rare and threatened plants. Okay, this is enough for sacred groves and biodiversity hotspots importance. Next question, again 3 marks question. It is, explain giving one example how co-extinction is one of the causes of loss of biodiversity. List the 3 other causes also without description. So we will see first co-extinction what it is. See, when a species getting extincted, that species to that, if any other species of organism showing obligatory type of relationship, or the organism which is associated with another organism in a obligatory way, they also will undergo extinction. Okay, this is majorly seen in symbiotic association as well as in parasitism. See, 2 examples it is given here. A host fish species become extinct. Its unique assemblage of parasites also meet the same fate, because they are losing their host. Because of that, this parasite is also undergoing extinction. So this co-extinction, it is occurring in parasitism in this way. And the co-evolved plant and pollinators, means the plant and pollinators they are there in mutual relationship. where one group, if it undergo extinction, the other group also will undergo extinction. That is also an obligatory relationship, but that is a mutual relationship. So here co-extinction means when a species become extinct, the other species which is in obligatory association with this extincted species also will undergo extinction. That can be seen in parasitism as well as in symbiosis and all. Parasitism, where when the host organism is undergoing extinction, the parasites also will be extincted. Host fish species becoming extinct, with that the parasite which is also extinct due to the host extinction. Another example, the co-evolved plant and pollinator means the plant it will co-evolve according to the pollinators, it will co-evolve according to the plant. Both the direction it can undergo co-evolution. So one, if it is undergoing extinction, the other also will be undergoing extinction. They are saying that they are in the mutual relationship. Okay, and next question in that, list the 3 other causes also. See, another cause, it is alien species inversion, and another cause of this this one, extinction of species, that is habitat loss and fragmentation. This is the major cause of extinction of the living organism. Okay, next one, over exploitation. So habitat loss and fragmentation, over exploitation, and this one, we can see alien species inversion and co-extinction. These are the different reasons for the extinction of living organisms. Next question, it is 5-mark question. It is an application level question. We will see that. It is with the choice. Okay. A heavily bleeding and bruised road accident victim was brought to a nursing home. The doctor immediately gave him an injection to protect him against a deadly disease. See, if any accident happened and if our body or wound it is exposed to the soil, there is a chance of getting the infection, tetanus. So immediately the injection which is given, that will be, what did the doctor inject into patient's body? Anti-tetanus injection will be given to the body. So first question answer is anti-tetanus injection. How do you think this injection would protect the patient against the disease? Because anti-tetanus injection, that is a ready-made antibody injecting to the body if any tetanus causing bacteria has entered inside the body, it will be eradicated. Now, name the disease against which this injection was given. Tetanus. And the kind of immunity it provides, that is artificial passive immunity is provided in the form of ready-made antibody. Now, next question they asked, for an organ transplant, it is an advantage to have an identical twin. Why? Organ transplant can be rejected by cell-mediated immunity in the body of human. So, if the organ transplant is occurring between the identical twin, they will get the organ which is isograft, means same type of organ, because identical twins, they have the same genetic composition. Because of that, whatever the matching, maximum matching in between the transplant or in between the organ of two individual, that is identical twins, it will be maximum. So we can say for organ transplant, the identical twin, it is selected because of their genetic similarity. Because of that, they will be having isograft. There is a very less chance of rejection of the organ by cell mediated immunity. Okay, so this is the way we can answer for this question. What did the doctor injected to patient's body? Anti-tetanus injection. How do you think this injection would protect the patient against the disease? Because this injection, it is a ready-made antibody injecting into it to against the tetanus causing bacteria. Name the disease against which this injection was given. Tetanus is the disease against that the injection was given, and the kind of immunity it provided, that is artificial passive immunity. And for an organ transplant, it is an advantage to have an identical twin. Why? Identical twin, they have isograft or similar type of organ because of the genetic similarity. So the rejection of organ transplant will be very less chance is there. as they will be showing more compatible as compared to other individual unrelated individual. Okay, so this is the way we can answer for this question. Next one, your advice is sought to improve the nitrogen content of the soil to be used for cultivation of a non-leguminous terrestrial crop. See, which type of bacteria, nitrogen fixing bacteria, we can introduce to improve the nitrogen content of the soil in a non-leguminous terrestrial crop. See, recommend two microbes that can enrich the soil with nitrogen. See, there are many free living nitrogen fixing bacteria, that is acetobacter, azospirillum. These are some example for that, and we can see some anabina, nostoc. These all will do the nitrogen fixation in the soil in free living form. And mostly acetobacter and azospirillum, they will do the nitrogen fixation. Why do leguminous crop not require such enrichment of the soil? Leguminous plants already they have symbiotic association with the rhizobium bacteria. Rhizobium, that is a good nitrogen fixing agent. Mention the role of cyanobacteria as a biofertilizer. Cyanobacteria like nostoc, anabina and all, they are capable of fixing the atmospheric nitrogen in the form of ammonia because they have nitrogenase enzyme. So the cyanobacteria, if it is present inside in the field, in the crop field, that increases the richness of the soil with the nitrogen content. Okay, that is the role of cyanobacteria acting as a biofertilizer. Okay, so this is all about this question paper.